Welcome to video 4 in our series on driving the power of single-cell technologies forward into the clinic with SkyVision. I'm Sarah Carl, Director of Data Science at Scailyte. In the last video, I described how we retain single-cell resolution in our analyses by evaluating individual cells with trained networks to score their predictiveness, and how that enables unbiased and sensitive discoveries. Today, I'll be talking about how we use explainable AI methods to extract the most relevant features from trained models, thereby allowing us to derive a simplified panel that could be used to prototype more clinically translatable assays. As we've seen, the combination of high-resolution single-cell omics data and targeted analytical approaches like supervised representation learning can lead to unparalleled discovery power, especially in the context of biomarker discovery. However, the biomarker signatures that come out of our discovery workflow are still very complex, and at least for now, it's hard to imagine implementing a single-cell assay as part of a standard clinical diagnostic or CDX. Therefore, we're interested in simplifying the signatures we find to the greatest degree possible in order to translate them to a more conventional assay, which could be applied in the clinical setting. We also believe that it's really important to be able to know which features our models used to make predictions, because this turns them from a black box into something that we can understand and builds trust among biologists and clinicians. There are a number of methods for so-called feature attribution available, and we've chosen to implement one called integrated gradients. The advantages of this method are that it's very computationally efficient and therefore scalable compared to other methods. It can be applied to any differentiable model without needing to modify the model, and it has some strong theoretical underpinnings, which you can read about in the original paper linked here if you're interested. To give just a general idea of how this works, you've probably seen this type of map, called a saliency map, for image data. Here we have a picture with the true label elephant, and a model was asked to predict what is in the picture. You can see on the left that it correctly predicted elephant with the highest probability, 0.55, although plural elephants was a close second. The attribution scores on the right show which pixels in the image were most important for making this correct prediction. And this is a local method, meaning that the values it calculates are specific to each input sample, or image in this case. Here's an example of how we can apply this method to a network trained on single-cell data. In this analysis, we collected publicly available data from multiple cancer types, comprising both primary tumor samples and brain metastasis samples that originated in those primary cancer types. We trained a series of models to predict whether samples were from a primary tumor or a metastatic site using a 6-fold cross-validation setup. And you can see here that overall they performed quite well. Then we selected the best performing models, which are in the upper right quadrant here, and applied integrated gradients to get the most highly predictive features from those models, in this case, genes. We rank them here by average attribution score, and note that these are averaged across both samples and models, so we get a pretty robust overall picture of which genes are the most important. If we wanted to dig into one individual sample, maybe one that was mispredicted, and understand why it was predicted that way, we could also do that, but here we're mainly interested in defining a set of genes that's common across samples. By the way, if you're interested in reading more about these results in detail, you can scan the QR code here to access our preprint on bioRxiv. Next, we took the top 20 genes according to the integrated gradients analysis and defined that as our brain metastasis signature. Then, of course, we could go back to the single-cell data and ask which cells express the signature. And we see here that it's expressed across multiple different cancer types, and primarily, though not exclusively, in the metastatic samples, which you would expect. Just to note, this dataset was subset to epithelial cells, but when we went back and scored all of the cell types in the dataset, we saw also high expression in other cell types, most notably endothelial cells. But again, it's hard to imagine developing a clinical assay based on single-cell gene expression, so we wanted to see if we could translate this to another data type. And we actually found that we could detect all of our signature genes in bulk RNA-seq data from tumor-educated platelets, so a blood-based sample. These platelets don't express endogenous mRNA, but they've been shown to pick up RNAs from their environment, notably from cancer cells. And what we found really exciting was that the signature was significantly more highly expressed in tumor-educated platelets from patients with a brain metastasis compared to patients with only a primary tumor. This raises the exciting possibility that one could develop a non-invasive assay to monitor brain metastasis, either in terms of progression or response to treatment, and maybe, although it needs to be validated, even for early detection of a metastasis before it shows up on a scan. In a clinical setting, discovery power matters, but so does translatability. And we have shown that our approach can enable this by developing multiple simplified assay prototypes in different indications. In the next video, which will be the last one in this series, I'll be putting all the pieces together and showing an end-to-end example of biomarker discovery, interpretation, and clinical translation in endometriosis. Hope to see you there.